Our webinar today is entitled Precision Oncology, Combining Orthotopic-PDX Models and MRI to Move Research Forward and Improve Clinical Outcomes. Today's webinar will be given by Jonathan Nakashima, the Chief Scientific Officer at Certis Oncology Solutions, based in San Diego, California. Jonathan has been working with patient-derived xenograft tumor models for over 15 years, starting this work during his PhD and postdoctoral fellowship at UCLA. He joined Certis back in 2018 as the third employee and has worked diligently to implement translational oncology assays for drug development as the company's grown over the past 3 years. During today's webinar, Jonathan will talk about orthotopic patient-derived xenograft tumor models and their importance to Certis's approach to studying drug efficacy in precision oncology. Jonathan will talk about some of the imaging modalities used at Certis for their in vivo studies, focusing mostly on the M3 system from Aspect Imaging and how the system has advanced their screening capabilities and progress in using the orthotopic-PDX models in their work. Before we begin, I'd like to briefly mention a few housekeeping items. We do anticipate today's presentation will be close to 30 to 35 minutes. This should leave us plenty of time at the end to answer any questions that you may have. Please use the Q&A dialog box at the bottom of your screen throughout the presentation, and I'll work with Jonathan to get through as many of those questions as we can during the hour that we do have together. We will create a transcript of the questions we answer during the session, as well as those that we may not have time to address during today's webinar. We will distribute this document within the next few days to the next week. Please also note that we are recording today's session, so if for some reason you lose connection or you can't hear everything clearly, you will have access to the video file later to review and share with your colleagues. So now, without further ado, I'd like to pass things over to Jonathan for today's webinar.
Great, thank you, Tanya, for that introduction, and thank you for Cintica for having me and for everyone attending this webinar. As you can tell by Tanya's introduction and by my title slide, today I'm going to talk a little bit about precision oncology and its multiple platforms, in particular how Certis is using the in vivo platform and combining that with imaging to discover or determine which therapies to provide to patients. I'll go through a couple case studies and then we'll wrap up with Q&A. Okay, just trying to advance my slide. There we go. So over the past 20 years, the good news is that there have been an increased number of drug approvals by the FDA for the treatment of cancer. And as you can see, there have been thousands of new oncology therapeutics that are in some stage of development. So there are a number of increased choices for drugs to treat patients with. But with the problem with that, with the more complexity, the more options becomes the more complexity. So this has given rise to a practice known as precision medicine or precision oncology. If you go to the NCI website, you can learn a little bit more here, but briefly what this summarizes saying is that each individual is unique in their own genetics, therefore their tumors are unique as well. And the information from those genetics can determine which plan of treatment and to figure out how that treatment is working and make prognosis off of that. This is in contrast to what's been done in the past, where patients have been given the standard of care. This is a list of chemotherapies, and if the patient fails the first drug, they go on the second. If they fail on the second, they go on the third. In this case, the patient really is the test subject, where nowadays, at the forefront of precision oncology is genetic sequencing. So this is the practice of looking at the mutations that make up a tumor to allow the oncologist and inform them on what type of drugs would best be suited for that patient. The advantage of this is that you can take a small biopsy from a patient, or you can even take a surgical resection that has been placed in formalin and do the sequence of that. And even nowadays, you can take blood and look at circulating tumor DNA to make an informed decision on what are the mutations that are perhaps driving the tumor. But as you can see from this review from a few years ago, the application of patients with metastatic cancer, only 15% of those patients have been received some sort of genetically guided therapy. And unfortunately, only about 6 to 7% of them actually had a clinical benefit from that. And I can say here at Certis, we have had patients who've come and had their tumor sequenced and have had results come back to them, and they listed, half a dozen to a dozen mutations of the tumor. And unfortunately, the results said that no drugs were available for that indication or that type of mutation. So the genetic sequencing is great, and again, it's at the forefront, and it's really driving precision oncology, but it's not a functional assay in that it doesn't quite bring us to the end and find the right answer. The next step of precision oncology is an in vitro screen. So this is taking a patient's tumor, a live tumor, growing it in a dish, like a petri dish, and then treating it with an array of drugs. Now, over the years, there have been tremendous advancements from going from 2D cultures all the way to 3D cultures, or now organoids. And this is the case where the tumor microenvironment is still present. So these 3D organoids contain the tumor cells, the stromal cells, and even in case the immune cells. So we can get a little bit more complex, a more representative model of the tumor. And then the advantage of this is that it's relatively fast to treat within a matter of days to weeks. It's high throughput because you can test a number of drugs simultaneously, and it's fairly inexpensive. The disadvantage of this is that it's still in a dish, it's on plastic. Granted, these organoids are floating, and again, more advancements are made to make it more physiological relevant. But at the end of the day, it's still in a dish. These organoids are being treated with drugs 24/7. The conditions such as the oxygen and the CO2 levels are vastly different than what happens in the body. But again, it is a vital platform for precision oncology. Lastly, and this is the area that Certis focuses on, is the in vivo testing. So this is where you take a patient's live tumor, we implant it in mice, and simply put, we expand the tumor, and then we treat the mice as if they are mini patients. There are a number of considerations to take when conducting an in vivo experiment, such as the strain of mice you're going to use. Now, you need to use an immunodeficient mouse, so that way the mouse's immune system does not reject the tumor. And there are a number of different strains and different types of mice that have differences in immunity. We use the nog mouse from Taconic because it is one of the most immunodeficient, but there are other strains out there, such as the NSG or other models, such as the RAG2 knockouts. Another consideration to take is the implantation technique that you will be utilizing for this. Now, you can take a fragment of the patient's tumor and place it in the mouse. And the advantage of that is that you retain the tumor microenvironment because you're taking a whole piece of that tumor and planting it. The disadvantage of that is that as you section up, some parts of the tumor is going to be different than the other part of the tumor. So you need to implant overage so that makes sure you get a good representation of that tumor. The opposite of that is to dissociate your cells and inject them. The advantage of that is that you get a much more homogeneous inoculation, and therefore each mouse gets a more representative distribution of the cells. However, the disadvantage of that is the dissociation of that tumor itself. Not only will it change the tumor cells and the gene expression of the tumors, but you lose a lot of those tumor cells, and then you go through an increased selection rate. So what we do at Certis here, we do focus mainly on the fragment implantation. However, we also do some single cell inoculations, which I'll explain a little bit later. And the last consideration, which I'll focus on more, is the site of implantation. So traditionally, patient-derived xenografts have been done in a heterotopic fashion, subcutaneously. This is where the tumor is implanted into the flank of a mouse in the rear, underneath the skin. The advantage of this is that it's much easier to implant, In fact, if you're doing a cell injection, you don't even have to anesthetize the mouse. So because this is much more higher throughput, and of course, because the tumor is growing on the back of the mouse, it's easy to monitor because you can use calipers to measure. So you can see if a treatment's working or not. The limitation of this is that it's obviously an improper tumor microenvironment. As you can imagine, a brain tumor growing doesn't grow in the back of a person under the skin. So the microenvironment is vastly different. And over the years, studies have shown how important the tumor microenvironment is for tumor biology. On the other hand, you have the orthotopic implant, and this is where Certis focuses on. The orthotopic implant is simply the Greek word for correct, and that means if a colorectal tumor is to be placed in the colon of a mouse, or a brain tumor in the brain. Obviously, this is a much more relevant microenvironment, and therefore is more biologically relevant. The limitation of this is that it requires technical skill to perform these surgeries. And of course, you need an imaging platform because the tumors aren't growing on the back of a mouse. There's been a number of publications showing differences biologically between subcutaneous and orthotopic implants. But perhaps what's most important, most striking are these two references I've listed here, is that the site of which a tumor is implanted can actually affect the response to therapy. So in order to become the most clinically relevant, we believe that the orthotopic model is the best model. And if you look at the history of successful drugs in clinical trials, you see that there's an over 90% failure rate in clinical trials. We believe that perhaps one of the reasons this is because the subcutaneous model is improper. But as I mentioned, one of the limitations of the orthotopic model is that you need some sort of imaging platform because these tumors are not growing in a fashion that you can easily monitor. So there are a number of in vivo imaging modalities, and at Certis, when we started, we considered all of them. Perhaps the most widely used in preclinical research is the optical modality, and this is where luciferase-based or fluorescence-based, where cells are tagged and you can monitor it by the light that it is giving off. The advantage of this is that it's really highly sensitive, so you can look at metastasis or look at distribution. And it's very high throughput. Most of the optical imagers can do 3 to 5 to 10 mice at a time. However, as I mentioned, the limitations, one of the main limitations, or I should say disadvantages, is that you have to tag your cells. And anytime you tag a cell, you're going to change the biology, and also you're going to go through a selection process. The other modality is a magnetic MRI, or magnetic resonance imaging. This is great for high tissue contrast. It has great spatial resolution, but the acquisition times are a bit long and it's low throughput. Right now, you can do 1 mouse at a time. The other options, ultrasound, also is very good, pretty high throughput as well, but there is limited depth and penetration on ultrasound, and it is a little bit more expertise dependent. We tried using some ultrasound, and it was just a bit of a steeper learning curve to determine on what was a tumor and to monitor tumor growth from there. And lastly, we have CT and PET, also great imaging modalities, but the disadvantages of that for CT was, one, the radiation exposure. You have also poor tissue contrast. And for PET CT, the short-lived radionuclides, one, you need a local chemistry facility to create your PET probes for you. With all this taken in mind, we chose to use MRI, mostly because for our patient studies, we did not want to perturb the tumors by making them into optical imaging assets. However, I will say that we are starting to do that for our preclinical research in here. We elected to go with the Aspect M3 as mentioned earlier in the intro, and below is just an example of the power that this machine can perform. So real briefly, I'll walk you through our methods at Certis. It's a simple process with many complicated steps in between. The step one is initiation study. So first we get a patient's tumor and we implant it into a small cohort of mice. We needed to first determine if the tumor is going to grow or not. If it does grow, then we passage it into a number of other mice, and we then grow those tumors. We monitor the tumor volume by our MRI, and when the tumors are of appropriate volume, we will randomize those mice and then treat those mice with the type of therapies that are prescribed by the oncologist. As an example for our orthotopic implants, is a snapshot of the different type of PDX and implants we have viewed by MRI. You can see here off on the left is our kind of our GI area where tumors are implanted into the liver or the gastric or in the colon. In the case of sarcomas, we implant it into the abdomen or the peritoneum, or if it's a bone tumor, an osteo or an Ewing's, we'll put it into the femur. On the bottom, you can see that we could also implant into the ovarian. You can monitor the pancreatic development, lung tumors, and as well as the soft tissue for sarcomas, and as well as the brain for glioblastoma. I've already mentioned that the tumor microenvironment plays a key role in tumor biology, and it is lacking in the subcutaneous modality. On the left here is histology from some of our models showing that you do get interaction between the tumor cells and the microenvironment. You can see here that a tumor injected into the bone degrades the bone and invades into the soft tissue. Another example here is the lung invading to the normal lung parenchyma, as well as the brain. So the brain is a glioblastoma and it is invading into the normal parenchyma of the brain. We also stain for human nucleoli to show that these tumors are still of human origin. And you can see the interaction of the tumor cells with the mouse stroma. As I briefly mentioned, one of the advantages of having a proper tumor microenvironment is that it can facilitate metastasis at a much higher rate. Here's an example on the right-hand side of a lung tumor that had spontaneously metastasized into the liver of this mouse. And with some 3D reconstruction, you can see how these metastases are spread throughout the animal. Now, as I mentioned, there have been a number of studies showing differences in subcutaneous and orthotopic, but we here at Certis wanted to see what we could figure out and determine. We took one of our colorectal PDXs, and this came from a patient's liver metastasis. And we simply put it head-to-head, subcutaneously and orthotopically. So this patient originally responded to the FOLFIRI regimen, and you can see the treatment regimen that we applied to our mice down here on the lower left. But by and large, you could take away that there was a striking difference in efficacy between the subcutaneous and orthotopic models, modalities, even though it is the same model. We elected to use MRI to monitor the subcutaneous tumors, even though you could do it by calipers. We used MRI to make it to be as non-biased as possible. You can see here on the subcutaneous model that there's very little effect between the control and the two treatments. And more importantly, you can see the histology is very similar across all three. Whereas in the orthotopic, we do see an effect with the FOLFIRI regimen. As I alluded to with the histology, I think it's probably more striking is that we see differences in histology between the control groups and the efficacious FOLFIRI group, with the tumor being much more differentiated, and there's a clear delineated border between the tumor and the liver. I think also if you just look at the controls, compare the control histology between the subcutaneous and orthotopic, you get a different morphology, with the orthotopic being control histology being much more de-differentiated and less differentiated. Lastly, what we also did was take these tumors and run gene expression analysis on this. And we found that the difference between subcutaneous and orthotopic, there were almost 350 genes differentially expressed. And when you subgroup these, we found that a majority of them fell into G protein coupled receptor signaling pathways, development, and most importantly, immune response. And I think this is going to be a real key factor with the emergence of cellular therapies and immunotherapies, where simply putting a tumor in a different location will affect how cells, immune cells, can be recruited to there. So with this in mind, I'm going to take a break here and pass it to Tanya to apply the audience poll.
Excellent. Thanks, Jonathan, so much for the great discussion so far. We would like to ask a couple of quick questions. So popping up on your screen now, you'll see a few questions. Just wondering which imaging platforms you're either using or would be best suited to meet your research needs. And if you are looking at specific cancer therapeutics, what are the types of therapeutics that you're focusing on? Is it small molecule inhibitors, checkpoint inhibitors, etc.? So please feel free to select as many options there as are appropriate for the work that you're doing. Also, as we continue with the webinar, please feel free to use the Q&A dialog box at the bottom of your screen to ask any questions of Jonathan about his work, the M3 system from Aspect Imaging. And again, at the end, I'll work through those with Jonathan in the time that we do have remaining. So we'll give everyone just a couple more moments to answer those questions for us. And thank you to everyone who's already taken the time. Perfect. So at this point, I will end the poll questions and I'll turn things back over to Jonathan for the rest of the webinar.
Okay, let's see if I can advance.
Perhaps left click on your screen, Jonathan.
Yep. Okay, there we go.
There we go.
All right, so now I'm going to talk about the clinical applications, how we're actually applying these for our patients. Here's an example of one of our most recent patients. As you can see here is a 47-year-old female. She had a leiomyosarcoma, multiply recurrent, resistant to pretty much everything, and even gone through some clinical trials, which she failed. So she came to us in need of help. And interestingly, she did have her tumor sequenced. And one of the top results that came out was a PARP inhibitor. So we started designing a study around a PARP inhibitor. And with additional, we had additional mice, so we put in other drugs as well. And when we started the study, we found that the NGS results, or at least the suggestion of a PARP inhibitor, turned out to be not true for her model. As you can see here, none of the olaparib groups were able to respond. However, we did find a therapy, a combination of gemcitabine and an HDAC inhibitor, to be very effective and actually induce some tumor regression here. On the right-hand side, you can see the summary of the results. Again, this tumor was implanted into the abdomen of the mouse. You can see the, I summarized the chart here, so it's not all the drugs because that would be a lot of figures. But you can see in summary that proof that the gemcitabine and the mosatinostat is the most efficacious. And again, histology, we always confirm with our patients on a histological level. You can see that not only do the tumors shrink, but the most effective treatment has the most differentiated phenotype and less mitoses as well. This next example here is another sarcoma. Sarcomas are very rare and there's not a lot of treatment options for them. So they make they are of a tumor type that could mostly be used using in vivo models to select their therapies. This example here is a pediatric rhabdomyosarcoma. There were a number of treatment options that were selected by the oncologist here. Again, you can see the results here where we found actually a number of good therapies to treat for her. The most prominent one was also, again, that HDAC inhibitor, mositinostat, with chemotherapy, vinorelbine. Again, this was implanted into the right thigh of a mouse, and you can see with histology, somewhat a response, but perhaps not as impressive as the last case. However, nonetheless, this patient, the oncologist elected to put her on cyclophosphamide and vinorelbine, again, due to some clinical factors that he determined. And you can see that after, I didn't say how long this was, but I think it's about 6 months, she has no evidence of disease based on PET scan from here. Now, one of the advantages of an in vivo model is that when you get to this point here, you can continue to do some other types of studies or follow-on studies. So when we got a great response from these about 6 drugs, the question is, well, which therapy do you put her on? In this case, what we'd like to do was stop treatment, let the tumors grow back, and just see what tumors grew back and which grew back as fastest. And this allowed us to infer and use recurrence in the animal model as an indicator of efficacy. So we can further stratify which drug would be a better therapy for the patient based off of the recurrence in the mouse. My last example here will be another sarcoma. This one is a 50-year-old male with, again, multiple recurrent, resistant, came from his right forearm. We elected to put it into the thigh of a mouse just for technical ease. Really, if we put it in the forearm of a mouse, chances are the mice will probably chew off the tumor. So we put it in the thigh of the mouse, and again, we found a therapy that worked for him. As you can see on the right histology, not only do we have a shrinkage of the tumor, but we give virtually no evidence of a disease based on histology as well. However, we wanted to see in the same fashion, if we stop treatment, will it grow? Oh, also, he has responded to this, so he's continuing on this treatment as well. But as I mentioned, what we did with this model, we decided to stop treatment, let it grow back, but in this time, we decided to treat it again. And the thought behind of this was to create a resistant model for, one, to get ahead for this patient should he recur, which we hope he doesn't, but we'll have something in play that we could quickly test. And 2, to create a resistant model, because resistance is one of the leading killers in metastasis of patients. And we know now that with PDXs, especially kind of in the research and development mode, some of the shortcomings is that they aren't as relevant to the current therapies. So we elected to create a resistant model off this patient's tumor, and it's something that we are continuing to do with some of our other models as well. So in summary, I've talked about precision oncology and the different platforms. Certis is focused on the in vivo model. I think, though, however, that we need all 3 platforms, the in silico, in vitro, and in vivo. And this diagram adapted from this publication is a good explanation of how all 3 can work. I'm excited to say that we are creating tumor organoids or spheroids off these lines. However, it's not quite ready, and we're still focused on the in vivo model. One other thing too is that the limitations of the immuno, because that they are immuno compromised, it is difficult to do immunotherapies in these animals. However, with access to patient tumors and tissue, we are now collecting and culturing the tumor infiltrating lymphocytes out of these tumors, as well as collecting patient blood, with the hopes to create a more autologous tumor to test immunotherapies for patients, as well as to advance drug discovery as well. So with that, I will end here and just say thank you to the audience, as well as Cintica for putting this on. If you have any more questions, you can reach out to me or log on to our website. Again, I didn't really talk about a lot of our drug discovery and our drug discovery services, but we can learn more about there from our website. And with that, I will end.
Perfect. Thank you, Jonathan, so much for the great presentation thus far. There are a few questions. For those who have questions that they've been holding back, please do use that Q&A dialog box at the bottom of the screen. And we've got about half an hour or so to work through some of those questions, should they be there for you guys. So Jonathan, an interesting question. How much tissue does it take for you to, Jonathan, if you want to leave that last slide up, that'd be great.
Sorry.
No worries. How much tissue does it take for you to start one of the orthotopic PDX models?
That's a good question. So obviously, the more is the better, because we can do more things with it, and we can also create culture with that. But to create a simple model, we've actually been successful with pleural effusion. So we took a patient, I think it was about 300 mils of pleural effusion. We spun it down to a cell pellet. It was, I'm forgetting the number of cells, but it was really small. Traditionally, and what we're moving towards is core needle biopsies. For those who are unfamiliar with that, it's about, I think, a 14 gauge core needle biopsy. It's about the thickness of a spaghetti noodle, and maybe just about a centimeter of that. So we've been able to successfully generate PDX models from core needles. In fact, the pediatric case that I showed was from that as well.
Nice. Perfect. And then from, I guess, receipt of the patient sample through to when you're able to provide feedback to the physician, how long does that process normally take?
That's a good question too. It obviously varies depending on the patient's tumor. We've published in sarcoma that the median establishment was 54 days. So that's just for step one. And then step two is a process that takes the pharmacology itself takes about 4 weeks. We do a 4-week pharmacology study. Plus, in order to grow those tumors up to that size, it's about another 3 weeks. So we're looking at, what is that? 60 days plus another 4 weeks. So it's about, it's not, unfortunately, it's not as quick as the other platforms like in vitro. It's about, 4 to 6 months or so on average. Again, some are really fast, some are really slow. So
yeah, that makes sense. Another question about how does subcutaneous versus orthotopic tumor models, how does the treatment that you are using there, how does that translate to the actual treatment in the human side of things?
That's a good question. So we have a number of studies undergoing right now that we're just getting data back between subcutaneous and ortho. We're not quite concording that with the patient. It's more just to see what are the differences and do they respond differently. Again, I think what we're seeing here is every PDX tumor is different, where in the case I showed of the colorectal, that patient did respond to FOLFIRI, and the orthotopic model did respond. Subcutaneous did not. I think we're finding some of the models don't show much of a difference. Some models show quite a difference. What we are doing is NGS on those models, and I think we just got those results back and we're analyzing now, and we're seeing very similar kind of themes in that, and that there's differences in signaling and mostly immune response. So I think we're going to find that the orthotopic models, especially in the case of cellular therapeutics and immuno-oncology, it's going to be really prevalent to perform a orthotopic study versus sub-Q.
Great. A little bit more about the imaging modality. So obviously I've worked with you and the team on the MR system. Can you help me understand a little bit more about the throughput? For example, how long is the team spending on each animal to acquire the images that you were showing, for example, today?
Right. So it is a bit lower throughput, I will say, and my team, being the perfectionists they probably are spending too much time on that. As you know, the acquisition times can be as short as 1 to 2 minutes, all the way up to 8 minutes. So it really depends. We have a study that we're running in brain for a client, and they wanted to start really small, around 1 or 2 millimeters cubed. So because of that, we needed a longer acquisition time.
Right.
For our patients, because some of these tumors grow rapidly, and in the case of if we're doing a sarcoma where it can be a little bit larger and a little bit easier to to identify, it comes out to about, I'm gonna say 6 minutes per mouse or so. So it's a full day. It's not, it's definitely not um, as high throughput as we'd like, but I think the quality of the science overrides that.
Definitely. Moving back to the science a little bit more, one of the questions is, what about using tolerized mice models, allowing you to grow human tumors in immunocompetent mice? Do you see a future there?
Yes. Actually, I'm interested to learn a little bit more about that.
Mm-hmm.
I, I don't have enough information to give an appropriate answer,
Sure.
but I do know there is some work to do with that. I know in general there's also genetically engineered mice that have the human receptors, so you can still put in a mouse tumor, but use your therapies, or IO therapies against made for a human, because it's binding the human receptors. Tolerized for PDXs, I'm not quite familiar with that. Whoever
We'll connect you. Don't worry, Jonathan.
yeah,
Yeah,
that'd be great.
absolutely. Dr. Urs will share your information with Jonathan, and you guys can follow up perhaps and have a larger discussion about it. No problem there. Another question regarding the imaging. So what is the smallest tumor that can be detected using the MR, and specifically in millimeters, for example?
Great. So yeah, our brain work, we've gone down to 1 millimeters cubed that way.
Yep, that makes sense. I guess I'll expand a little bit on that. It's having worked with the team at Certis, as well as obviously the instrument for a few years now. The detection limit's going to depend on the resolution of the image that you're using. And so typical resolutions are going to be anywhere from 150 microns to 300 microns. And so you do want a multiple of that resolution to be confident that what you're seeing is in fact a tumor. So I would very much second what Jonathan said. Around 1 millimeter should be doable using the system. Excellent. I don't have any other questions right now. So if there are any people lingering out there, okay, one just popped in. Do you consider the differences in the gender of the mice when you're doing your studies?
Oh, that's yeah, that is a very good
Mm-hmm.
question. So we do, unfortunately, for the technicalities and the throughput, the majority of our work is done in female mice. This is because males will fight, and when you're running a large study and you have to randomize, by randomization, so let's say we implant 100 mice and then we image them and then we take the best 60, we will consolidate those 60 and then take the other 44 R&D. And in doing those mixing and matching, as you probably know, if you do that to males, they'll fight, and then you have to single house them, and that just drives up cost way too much. So we use female for the most part, unless we're doing like a prostate or something really kind of really gender specific. But for the most part, it's in female mice.
Perfect. Thank you. Do you resequence tumors after growing them in the mice in order to confirm that they haven't changed from the original patient profile?
Yes, we do, and they don't change much. I mean, obviously, you're going to get drift over time, but in our early passages, they don't drift much.
Nice. That's an excellent question. I'll give everyone a few more moments just to type in any questions that they may have. But I do want to thank Jonathan for his time in putting together the presentation today. I very much enjoyed working with the team and look forward to continuing working with them. And I thank everyone for their attendance today at the webinar. We do appreciate everyone's time and know that everyone's quite busy. So without any other questions coming in, please feel free to reach out to either Jonathan at Certis or myself here at Syntica, and we'd be happy to connect as appropriate, depending on the questions that are asked. So at this point, let's be respectful of everyone's time. We can all get 20 minutes back in our calendar, which I'm sure will be helpful. And again, I'd like to thank Jonathan for his time and hope that we've provided you with some interesting information about using MRI in precision oncology, specifically on orthotopic PDX models. if in the days and weeks to come you have additional questions, like I said, feel free to reach out to us at Syntica, and we're happy to discuss further or connect you with the team at Certis as well. So thanks again, and we look forward to seeing you at a future Syntica instrumentation event.